interaction in deep learning protein design models.
So, So first of all, I'd like to thank the organizers and colleagues in Nanjing University for inviting me here and to share something about de novo protein design models. So previously we have hearing so many different aspects of protein design. So I only need to start from here to say that currently de novo protein design methods are still evolving rapidly. But still, I don't know if it's fortunate or unfortunate, it still follows this mainstream. This backbone first generates the designable backbone and then optimize the sequence. So previously, before 2002, we are doing this, either generate backbone or design sequence with energy functions. But nowadays, more and more artificial intelligence generated methods are being developed. The current models for backbone generation, most successful one are diffusion models, and also hallucination. And also for the sequence design, it's basically mostly based on graph networks to encode the three-dimensional environment of the backbone and then decode it to residue types. You can do it either iteratively or through autoregression or projection. But this process, the focus up to now, I think, although people aim at design functional proteins, but the focus in this method development process are on the physical plausibility of the backbone and also the structural stability of the final sequence. And this gives limitations when people want to apply current method to functional protein design because of accurate modeling of molecular interactions, which are very important for functioning. And also, we have seen that protein dynamics are also important functions. So they are biased towards structures of high rigidity. So this leads to suboptimal performances in designing functioning proteins. So today, I will focus on a particular piece of recent work, which is only recently accepted in Nature Communications, and I'm grateful to that. So this is about inverse folding, given the backbone, and when we design how to design the sequence with the consideration of atomic interactions, not only the backbones and also the side chains, and also the interaction with target ligands, and also to prepare us for designing proteins with dynamics, try to also include input of multiple conformational states, and also for functional protein design, for enzymes, sometimes we cannot really fully understand where what are the constraints on sequence. We use integrate evolution information, multiple sequence alignment into this model. So the Abax-T compared to existing models, so the sequence are generated not only the residue types, but also use explicit model the side chain structures. And the name Abacus is for inverse folding, data-driven. And so I will have time to just delve a little bit deep into the model. So here we have different type of input, and the main network of Abax-T contains the encoder module, a decoder module, and iterator module. So the goal, the aim of the encoder module is processing the input conditions, and the decoder module is to translate into residue types, and the task of explicitly modeling the interactions our sidechains, modern sidechains are, and also with language models, we integrate language models to encode the sequence generated, are given to the iterator module. So this is actually a pretty standard approach now in encoding structure information. So we have backbone structures, and we use a neural network to encode the backbone. And we have ligand, and also use some pre-trained chemical networks to encode the chemical properties of the ligand. After this embedding, so we have interactions between the explicit interactions between the ligand and the backbone, and have this representation. Of course, this representation is updated with several layers of graph networks. And the decoder module of BuxT is we allow the model to be able to use either input or a single conformational state. So here we need to translate the encoded backbone and including ligand structures with the updating networks and projections into residue types. So we are using discrete space, state space to decode the entire sequence gradually. And also we have sidechain conformation decoders, so that all this can be sent to the iterator module, so that when the sequences are gradually optimized, they will see that the intermediate structures, all atom structures. And also we allow multiple conformations to be input into the decoder. So basically, this is adaptation from the single state structures. So we have, instead of one input structure, we have multiple structures. Some of them may have different ligand interactions. One may be the apple, the other the hollow state structures. And then after encoding and pooling, then we decode that into the same residue, amino acid residue sequence, but also we decode them into different sidechain conformations. And in the iterator module, we have integrated language models so that the residue embedding see some context of the sequence. And also we have express sidechain atoms interacting with the ligands and backbones. And this, after encoding, were sent back to the diffusion decoder as part of the input, so that as self-conditioning. So we show that these different components, so we developed this by heuristic trial and errors and try to improve them. We show that these different components contribute to the improved accuracy of BuxT. For example, the top row is the full model with everything. And here we gradually reduce the scale, the size of language model, and we see gradual dropping of the native recovery rate. And without the iterator, without the language model and the sidechain conformations, also the deteriorating of the recovery rate, and without the ligands, we see very significant deterioration of the pocket residues. And also we see that the language models, because here we tested on native proteins, they really capture the active site residues. So if we have language model and given the native structures, they recover the active site residues very well. And if we use this long iterative single step projection, we get the worst performance. So the largest improvements are for the ligand interacting residues, which means that the model has somehow captured the interaction with the ligand when trying to design the sequence. So we compare this with existing models for inverse folding. So here we have a sequence of models from earlier ones, all the different methods, with a graph neural network type of approach. With more recent ones, we see the gradual improvement until BuxT. Indeed, in terms of native sequence recovery rate, BuxT generally the highest metric. And also in terms of structural properties. So here we send the design sequence to ESM fold. No, this should be AlphaFold 2, and they look at the structural quality of the predict structures, and the quality is also among the top performing models for inverse folding. And also we check the performance of the model for different ligand types. For example, nucleic acids, small molecules, ions. So the red one is a box T, and the others are different models. Some of them don't have ligands. And if we look at the side chain quality predicted by a box T and compare them against side chain packing state of the side chain packing models, a box T also seems to be performed very well. And so if we compare with the native sequence, so we redesign the sequence on native proteins, and we apply the structural prediction programs to it, for example, ESM, it's interesting to see that the design sequence actually contains more information about the native structure than the native sequence, which can be more easily better captured by structure prediction methods. And if we look at the energies, we take the structure and optimize the structure and calculate the loss energy and compare with the energy associated with the native sequence. We see an improvement of total energy for most of the redesigned proteins, and also improvement, most significant improvement in the van der Waals energy, and some improvement in the statistical improvement in the electrostatic energy, even though we did not explicitly model in the energy. So this slide will show the effect of considering multiple backbone states. So we collect a set of benchmark proteins with multiple experimental structures, and we compare multiple state design and the design on each state, and also a model trained on only a single state. Again, we see that for these proteins, with the model considering multiple states, gives the highest recovery rate of native sequences. And also, we have another model which is trying to predict, we call it PVQD, to predict a conformational ensemble from a single sequence. So we apply this model called PVQD to all these sequences and look at the correlation of the predicted structure distribution, the RMSF correlation with the experimental observed deviation, and also the time score, TM score to the most similar experimental structures. We also see consistent improvement with these multi-state design models. So how about experimental test? So to test this multi-state design capability of ABACS-T, so we choose a simple, relatively simple protein with a very simple conformational change, allose binding protein. So this naturally, it has the open state without the ligand, and it has the closed state with allose bond. So we try different design methods. So we will not summarize, compare different methods here, but I only tell the results is that with this multiple state design approach, we were able to design 7 sequences. So just 7 sequences, just one shot design, takes the design, and the sequence identity to the native protein is about 75 to 80%. So 50 to 60 mutated residues. And all these design proteins, they show binding with allose with affinity from 1/10 of the native to 17-fold of the native protein. And we solved the crystal structures for 2 proteins in the upper state and the hollow state. And we'll see that the design protein indeed has the ability to change its conformation upon binding to allose. And also because we are doing structure-based design. So the design proteins, this strong high-spanning affinity protein, and also the ones showing the experimental structures, and we characterize its thermal stability, and we see an increase in melting temperature in 10 degrees. Of course, we have already seen that if we apply structure-based immerse folding to functional proteins such as enzymes, because we obtain proteins are highly structurally stable, but most often they were functionally inactive. Of course, one of the problems is molecular interactions and conformational dynamics, and also there's something we don't know about the enzyme catalysis. So here we try to adapt this method for the zero-shot improvement of enzymes because it is so effective in designing stability-enhanced proteins. So we try to integrate for natural enzymes with multiple sequence information, with MSA transformer, and then with this model, without fixing any residues, just let the model run, and first, of course, we test this method on a protein data set where we have different mutants and we have activity data, and we show that the Spearman correlation of different methods, we show that with ABAX-T and with MSA, we indeed could improve the Spearman correlation to this existing data. So we test it on real enzymes. So this is endo-beta xanolase. So we apply ABAX-T without the MSA information, and we get the results in red. This is the activity, and we see significant loss of activity. But if we incorporate the MSA information, from a few tested proteins, we get proteins with wild-type activity, and also this protein is thermodynamically very stable, so it can resist to treatment at 80 degrees, but the wild-type is lost activity after 10 minutes. And also this protein can tolerate to other adverse conditions, probably because of enhanced structural stability, like high pH and other harsh conditions. So we also tested on another enzyme, 10-beta lactamase, and in this case, without the MSA information, we still see some loss of activity, but with MSA information, we can actually improve the activity of the enzyme with dozens of mutations, and also significantly improve the blue and the red, the thermal stability of the enzyme, and in one shot design. We also tested it because here one focus of ABAX-T of interaction with the ligand. So we test this oxylactamase. It naturally can work on several different substrates. So we see if we can redesign the substrate selectivity of this enzyme. And so these are the different substrate of the protein. And actually, these are the selectivity showing the redesigned enzymes compared relative to the wild-type activity on different substrates. We see that indeed we can shift the activity for these two ligands. So we are not 100% successful, but we can partly shift the substrate selectivity. Again, we see significant improvement in thermal stability of all the redesigned enzymes. So if we look at the redesigned proteins, compare the mutants or the thermodynamically improved enzymes and calculate the energy, because people are interested in why these designs enhance the stability. And we see that for most of them, indeed, the calculated energy, Rosetta energy per residue, or the total Rosetta energy, total Rosetta energy are indeed lower than the wild type. And if we distribute this total energy difference to individual residues, and we partition them, the contribution into two parts, a small number of residues with large energy contribution, and the large number of residues with small contributions, but when then add them up together, we see that for most of the designs, actually the larger, this part, the weak contributions from different residues are larger than this small number of key residues, single residues. So that's really interesting effect of this deep learning-based design, because if we compare the deep learning methods, different learning methods with energy score-based method for single mutants, because we have, on a database on the stability, we see that in terms of correlation with experiment data for single mutants, the energy scores still outperforms all these different deep learning methods for prediction of single mutations. So deep learning methods do not yet outperform energy scores on single mutations. But what's interesting is that the strength of deep learning, despite this inaccuracy, they seem to be generate high success rate in designing many mutants. Yeah. So this is perhaps one thing is that it indicates that there are still rooms to improve deep learning methods so that they get more physical, so that at least they can outperform the energy score method for these small perturbations. And the other thing is that with current methods, energy score, you can, perhaps I don't think you can optimize the energy score and get very large sequence deviation from the original and still get good results. But it could be a very good scoring method, judge method to evaluate results generated by deep learning method. Okay, so just a summary of our Box-T. So we have improved inverse folding accuracy by modeling all atom side chains and also allowing interactions with ligand, and also have the ability to treat multiple conformations, multiple states. Of course, the limitation is that this, as the inverse folding method, you rely on quality holistic structures, and currently from experiment. But we hope that we will be able to generate designable conformational ensembles instead of single structures. So my last slide, so I think the current state and key frontiers of generated de novo protein design, I think the problem of design structurally stable protein monomers and also design protein complexes related on relatively simple interfaces such as helices could be considered robustly solved. And we see a lot of talks about the closing to be solved protein complexes with intricate interfaces such as antibody-antigen complexes. And the major open challenge is controllable design proteins are controllable conformational dynamics. This including the designing of enzyme of high activity without large-scale screening and design proteins of programmable dynamic behaviors without relying on very smart heuristics. And with that, I end my presentation. So I would like to thank my group members, and the experimental test has been performed by Professor Chen Cheng and students. And also the deep learning models I talked about is developed by a very talented PhD student, Yufeng Liu, and in collaboration with Computer Science Department of USTC and also IFLATEC. And thank you. And
thank you for the very interesting and useful tools for the deep learning-based protein design. I think there's a question.
Professor Liu, great talk. So I'm wondering whether your method can be applied to de novo design protein. So you have shown that it can be applied to a naturally exist protein, which can improve its thermal stability and the enzymatic activity. So basically I'm wondering to what extent the language model play a role in that, and because for de novo design protein, probably you don't have a lot of related similar sequences in the database.
Yes, so we have tested on de novo design proteins. We have actually experiment tested with de novo generate backbones, because another direction of a group is for generating de novo backbones. It seems to be work well, but because the de novo backbones so far, they are much easier to design compared to native proteins. And also the language models, I think even for de novo proteins, because ESM is actually, I think it captures some, even for de novo sequence, de novo structures. Because all these de novo protein design tools, the models are still learning from natural backbones. So somehow the language models handle sequences for novel structures, even they still work. Yeah. ESM fold works for de novo design proteins. Yeah.
Great talk. Thank you.
Yeah.
Yes. So once again, we give the thanks for the brilliant talk. So the next session will be two flash talks. So the first flash talk is given by Professor Bin Shao. He's a professor at the Beijing Institute of Technology, and his talk will be introducing plasmid GPT. It's a generative framework for plasmid analysis and generation.
左边，左边。Okay, thank you, boss. It's my great honor to have the opportunity to speak here. Thank you very much to the organizers. And my name is Bin Shao, and I work on DNA language models, and I'm here to talk about a DNA language model for engineered plasmid sequences. So I brought the left image from Twitter. I asked Dimitri whether I can use this image, and he said yes. And it shows the airports around the world, the ports around the world, and the real roads around the world, and also the DNA language model users around the world. So it's black. It's a joke I work on DNA language models, but if we look at other models, like protein language models, they are widely used. I think Martin talked a little bit about it earlier today, is that for ESM models, it has some drawbacks, but it's very popular. So what might be the reason behind this difference? I have a possible answer, but before that, we can first look at the landscape for DNA language models. So we know that large language models has been very successful in modeling human language, like English or Chinese. And so people are using similar strategies to try to model like DNA synthesis or protein synthesis. So I think yesterday, Brian and Nolia, they have covered this topic very nicely. And on the left, it is a review by Professor Song Yun from UC Berkeley. And it is a very nice review, covers all the DNA language models. And in the middle, I highlighted a few works. The works from Brian's lab, EVO1 and EVO2, they are really pushing the field forward. And also from Professor Song Yun's lab at UC Berkeley, the GPN models. More recently, GPN Star is really making some difference. And also for the labs in China, Zhao Rongli's labs, they have developed Luca1 and more recently Genos. So Genos is a long context, single nucleotide resolution DNA language model for human genomes. I think that's what we need. And for our lab, we have developed MegaDNA before. So it's a long context language model for bacteriophage genomes. It was published last year. And I'm not going to talk about this work today. But if we look at all these DNA language models, they share one thing in common, is that they focus on genome synthesis. And that might be the problem, because for genome synthesis, we already have many very good tools for them. We have very good sequence-to-function models, like alpha genome, like Informer, like Parfin, and also the models Professor Yekai talked about yesterday. So you would really need to show some improvement compared to these existing tools, and I think there are some active debates about that. But there are also some DNA sequences that people are not talking about. So these are engineered plasmid sequences. And these sequences, they are publicly available. Thanks to the tremendous efforts of IDGene, they can send these plasmids to every corner around the world. And for myself, I constructed about 6 plasmids. I put them on IDGene when I was a postdoc at the Chris Watts lab, and they have been shared about 50 times to different labs across different continents. And also the number of engineered DNA sequences that are increasing rapidly. For every year, we have about 10,000 new plasmid sequences available. But despite this, the engineering DNA is still a very hard task. I've done molecular cloning for 3 years, and you will need to select your plasmid backbone. You will need to select the regulatory elements like promoters or terminators, and you will need to copy-paste your DNA sequences manually. And that's really labor-intense. So how can we make it a little easier? The large language models has been used to help us engineer DNA. So maybe 1 year or 2 years ago, Future House, they have proposed a lab bench, which is trying to evaluate how large language models can help us to do molecular clonings. And more recently, I think Belmani from Julia's lab at Stanford, and also CRISPR-GPT from Congle's lab and Mengdi Wang's lab, they are trying to use LLM agents to give us step-by-step instruction about how we can do molecular clonings. But despite these kind of developments, we still lack a language model that is specifically trained on engineered sequences, that can understand engineered sequences. For all this evaluation framework, all these agents, they use the large language model that was designed for human language. So that's what we did. We constructed a language model just for engineered plasmid sequences. We used a decoder-only structure, which is just the GPT model, and it used the next token prediction task to do the model training. It's quite simple. You predict what is the next word, what is the next word, so you can construct the whole paragraph. So it's a pretty simple model structure, but it works for us. The only 2 differences that we implemented is that first we use BPE tokenization. Before, in our previous work like MegaDNA, we use single nucleotide tokenization, but for plasmid sequences, there are some parts that people are reusing it again and again, so we use 1 token to represent those kind of frequently used parts. And we also use circular shift augmentation, is that for plasmids, they are circular, so we can choose our starting point at different positions on the plasmids. So after our model is trained, we are curious whether it learned anything meaningful about these plasmid sequences. So we look into the embeddings, so you can feed plasmid sequences into the language model, and the language model will produce a high-dimensional vector, which is called the embeddings. And we can also calculate the embeddings for each research lab. For all the plasmids from 1 research lab, we can calculate all the embeddings for the plasmid and do the average. So on the left, we plot the lab embeddings in a 2D space using t-SNE, and each dot corresponds to 1 lab. We can see a clear separation of different research labs. And also, we can see the labs are clustered based on what they are working on, whether they are working on mammal systems or whether they are working on bacterial systems. And we can dive into some specific labs. For example, we can look at the Chris Watts lab, which is the lab I worked before. And we can plot the embedding for each plasmid. We can also see the plasmid embeddings are clustered together based on the publication. So in the middle part, the color corresponds to different publications, and also they are also separated by publication year. And on the bottom, we also see the plasmid from Professor Feng Zhang's lab, and they contribute maybe thousands of plasmid to the IDGene database. We can also see a good separation of different publications. So that means our model actually learned something meaningful for different research labs and also for different plasmid sequences. So the next thing we want to do is to do some prediction tasks, is that whether we can predict some features for these engineered plasmids. If you look at the IDGene page for each plasmid, here I show an example from one of the plasmids that I have engineered. It shows which publication it comes from, and also what is an antibiotics resistant marker, and also what is growth strain. So there are rich information there. So whether we can use our model to predict those features. And the prediction we done prediction on the bottom. So is that we put the plasmid sequences into the language model, and we produce the plasmid embeddings, and we use those embeddings to predict where the plasmid comes from, and what species it comes from, and also the growth strain for those plasmids. And for the first task, it's also called the genetic engineering attribution, is whether you can find the origin of one engineered plasmid sequences. So that has some applications in biosecurity or biomonitoring efforts. So there are also already some works in that direction, so I list them on the bottom. So here's the performance of our model. We benchmark the performance of our plasmid GPT model against CNN models and also other existing models like plasmid Hawk. And we found our model shows improved performance. And we also look at how, for the lab origin prediction, we look at what factor determines the accuracy for this task, whether it is related to number of the plasmids from one lab or whether it is related to the diversity of plasmids from one lab. So it's on the bottom. And here are also two examples about the plasmid feature prediction task. Is that we have two plasmids that are not in the training data set, and we have the ground truth on the left, and we use our model to predict the features of this plasmid, which lab it comes from. And we also have a collab notebook that makes it easier for you to do this prediction. You can just copy-paste the plasmid sequences into your web browser, and it will do all the prediction for you. And last, we did some generation of plasmid sequences. Is that because we are a generative model, we can let the model to produce some plasmids that might be functional, and on the top part we have generative plasmids that can extract VIP stably in E. coli cells, and on the bottom we can also do some conditional generation of plasmids that we can fine-tune the model based on what kind of plasmid we want to generate, whether it is mammal expression or whether it is for lentiviral vectors. So that's my talk, and our work is on bioRxiv. Our model is publicly available, and thank you again for the opportunity. We sincerely appreciate the time, insight, and the constructive guidance of editors and anonymous reviewers to help us improve our work. And thanks so much for your attention, and I'm ready to take any questions.
Yeah. Thanks. Are there any questions? Okay, so if there's no questions, I will ask a quick one. So how do you validate the functionality of the plasmid you designed, actually?
So for this plasmid, it's not here, but the function is quite simple. Is to express VIP in bacterial cells. So you need to have all the necessary components, like resistance marker, like replication origin, like terminator for the VIP protein. So as long as it has all the components, I think it should be good. And we also do the Y lab experiment to validate that.
I see. Thank you so much. So the next talk will be given by Jun Xia from Hong Kong University of Science and Technology. And that talk will be based on LLM-powered MSA agent, transforms, tandem mass spectrum, annotation from manual to automated.
Okay, so first of all, I would like to express my gratitude to the organizers and Professor Ni's introduction and all the audience. So today, I'm Jun Xia. I'm an assistant professor at HKUST Guangzhou and HKUST. So it's my great honor to be here to present our recent work on behalf of our spectral AI team. Yeah, today my talk focus on the deciphering proteomic and metabolomic dark matters with AI foundation models. So as we all know, the human genomic project has been completed at 2003. However, the human genome cannot reflect the dynamic life status of humans because the human genome is relatively stable compared to the human proteome. Therefore, the human proteomic project has been studied recently, aiming to map the proteomic changes across the entire human's lifespan. So this internationally collaborative project has provided valuable data resource and intelligence support for our spectral AI research. So as we know, the AlphaFold series has achieved overwhelming success in protein structure prediction. But how can we obtain the protein sequence as the input for AlphaFold? So the most widely used tool is the mass spectrometer. However, the state-of-the-art tools can only achieve nearly 50% score in protein identification task, which is still far from real-world applications where we try to find the proteomic dark matters. So it's very similar to the protein structure predictions task before AlphaFold. On the other hand, despite the importance, the AI for science community hasn't paid enough attention for this important task compared to the protein design or molecular design and so on. So inspired by the AlphaFold series, we try to build the AlphaFold for mass spectrum-based protein identification. This is the specific pipeline of protein identification using mass spectrometer. So one of the important and most challenging tasks in this pipeline is peptide sequencing, where we try to translate the mass spectrum data to the peptide sequence. There are two paradigms for this task. The first is the database search, where we compare the orbits of the spectrum against the database and pick the most similar spectrum peptides as identification results. However, it cannot identify the unknown proteins out of the database, what we call them as dark matters in proteomics. The second paradigm is de novo peptide sequencing. It's very similar to the machine translation or image captioning in mainstream AI research. However, this task is much more challenging than these typical AI tasks because we have to predict the existing length of the peptide sequence. And also for each position, we have to know what types of amino acids in this position. And secondly, we know that there are 20 types of amino acids in nature. However, there are some variants called PTMS. So the current methods can struggle to identify these PTMS because they occur less frequently than other canonical amino acids. So to address these challenges, we developed a larger scale and general purpose foundation model for mass spectrum-based proteomics. So firstly, we connected the world's largest proteomics MS data, which contains nearly 145 million peptide and spectrum matches. These data come from different species, different enemies, and different instruments. This ensures our model can generalize to different cases in real world. And also, we built a model like a multimodal language model. So we feed the mass spectral data, regarding the mass spectral data as a sentence, and also the peptide as a sequence. And then we use contrastive learning to align the embedding space of the two modality. And also, we predict the peptide sequence conditionally. Here, we also incorporate some domain knowledge as the guidance for our method design. For example, we in a contrastive learning, we sample each peptides that share similar mass with the input mass spectrum as hard negatives. Then we conduct some hard negative mining strategies in machine learning to enhance the contrastive learning. And then we employ our ultra-prod model in nearly all the computational tasks in protein identification downstream tasks. Next, I will introduce this result in detail. So firstly, we activate the spectrum encoder, MS encoder, and the MS-based peptide decoder to conduct the task of de novo peptide sequencing. And we can observe that so our ultra-prod model achieved nearly 20% score, 80% score, which have outperformed previously best model by nearly 20% score. So this advancement is similar to the significant progress made by AlphaFold 1 in protein structure prediction task before. So we also observe that the ultra-prod model can achieve better performance in the data comes from human beings and the mouse. So which may be occur most frequently in our pre-training data. So our ultra-probe model can perform well in the downstream task of peptide properties prediction, including the coalition, cross-section prediction, and also the spectrum prediction, which can help us to identify more peptides in the traditional database search paradigm. It can also predict the theoretical spectrum accurately, as can be observed. The peptide properties predicted by our ultra-probe model are very close to the experimentally measured results, which can enhance the database search in the target case. So our ultra-probe model can cluster mass spectrometry data based on the corresponding peptide sequence, and it has significantly outperformed the state-of-the-art mass spectrum clustering tools claims. Based on this ability, ultra-probe model can share a light on the dark proteomics of a human's body. So in other words, it can assign reliable sequence for the repeatedly observed yet consistently unidentified mass spectrum or dark matters in human's proteomics. So as I introduced before, so ultra-probe model embeds the peptide sequence and the spectrum data in a shared vector space, where we can calculate the similarity between them directly. Therefore, we unify traditional database search and library search in a unified framework, and we name it as hybrid search. As a result, ultra-probe model can identify more PSM and unique peptides than state-of-the-art tools such as the mass quant and deep search. So based on these abilities of ultra-probe model, we built a streamlined protein identification workflow based on our foundation model. For a connected batch of mass spectrum data, we perform data hybrid search using the FDR metric. And for the left unannotated mass spectrum data, we activate different branches of our models to conduct de novo peptide sequencing to identify the left novel peptides. Finally, we collaborate with BGI and conduct some wet lab experiments in terms of immunopeptides to verify the effectiveness and useness of our ultra-probe model in real-world cases. So the immunopeptides here don't exhibit the characters from most proteomics experiments and cannot be identified by traditional database search tools. So we employ the ultra-probe model to sequence the immunopeptides, and then the BGI will synthesize these peptides and inject them into the mouse body to observe whether it can activate the immune response of the mouse. Also, the mass spectrum of the synthetic peptides are very similar to the naturally presented peptide sequence, which verifies that our ultra-probe model can boost the biological discovery in proteomics. So the next session is our MS agent for metabolomics. AlphaFold is known to all in the CASP contest. Similarly, there is a similar contest called the CASMI, in molecular structure elucidation from MS data. So, however, as can be observed, the best model can only identify nearly 30% metabolites from the mass spectrum because the molecular structure is much more complex than protein sequence. So many biologists hold opinion that the molecular structure elucidation remains in the pre-AlphaFold era. There are lots of challenges and opportunities. So currently, mass spectrum data analysis relies heavily on the labor-intensive manual work and specified expertise, domain-specific expertise. To address these challenges, we have developed the first LLM large language model-powered AI agent for MS data annotation and data analysis. So it integrates all the mass spectrum data analysis tools and combines the reasoning ability of large language models. So this enables it can automate the complete analysis process of the mass spectrum data, and it can also discover new metabolites beyond the scope of database. So we are also collaborating with some biologists to employ our MS agents in the discovery of new biomarkers of rare disease. So besides, we also develop an online service platform for the easy use of the beginners and biological experts for their metabolomics analysis, data analysis. So the last session is our outlook for spatial AI. So beyond the mass spectrum data, there are several other ways to elucidate the molecular structures, including the MR or infrared spectroscopy. However, the AI has seldomly been applied in these tasks. The final goal of our spatial AI team is to revitalize traditional database search paradigm and translate these advanced algorithms and models into practical use. Finally, I would like to express my gratitude to the talented members in our spatial AI team, and also the consultants and collaborators in science. So that's all of my presentation. Thanks again for your attention, and any questions is welcome. Thank you.
Thank you. Any questions? Okay, so I think we are a little bit behind schedule, so thanks again for your great talk. So the next session will be a panel discussion on topic of AI decoding life.
So let's welcome Angela, Martin, Mingyao, Nolia, and Robert for this short panel discussion. And we will talk about, actually this is just an open discussion, and we want to know what is the future for AI biology research, and also any suggestions for young researchers who are interested in joining this fantastic field.
Can take a seat according to this order. 1, 2, it's Ming Yao is here. Martin,
Julia.
Okay, yeah, sure. You may take a seat, and let me take this. So first, welcome everyone back to stage and join this panel discussion on AI decoding life. And also, I'm very honored to host this session with Qian together. So yeah. As Qian already mentioned, so in this session, we'll really welcome your questions regarding AI augmented biology, no matter them being general ones or specific ones about talks. And maybe I can start one question with a general question. So throughout the days, I have listened to many excellent talks, and I'm aware that now AI technology is basically integrated in all aspects of biology. So, but I also realized that for some really excellent talks that involve a lot of computational work, maybe it's a little bit difficult for conventional biologists to understand. So my question is, maybe as a computational scientist, do you feel that it's necessary nowadays for maybe pure biologists to know a little bit about bioinformatics, and this may be helpful for them to integrate AI technology when it's necessary for their research? And in a broader sense, maybe this is also helpful to minimize the potential gap between computational biologists and also, yeah, conventional biologists. Thanks. If anyone wish to answer this question.
I'm happy to go ahead. So I think every wet lab scientist should know a little bit of computation. I think that's really a critical skill these days. But should everybody be an AI expert? I don't think so. But I think also the computational community has a lot of responsibility here too. So I feel like we are really in charge to make our tools available and easy to use and really accessible. And I guess if we do a bad job at that, we will lose a lot, you know. And I think that it's double-sided. I think both sides but both communities have to be getting a bit closer together and yeah, making things accessible as well as people learn a little bit how the background of these methods really work.
Thanks.
So my name is Mingyao Li. I'm from University of Pennsylvania. I work with a lot of people, biologists and medical researchers. I work with different types of medical doctors, and they have apparently very different backgrounds. Some of the people are very savvy computationally and statistically, and they even know machine learning, and they can analyze the data themselves. And those people are much easier for me to communicate with because they can understand the sophisticated machine learning AI algorithms very well. But there are other people, they are pure biologists. They know very little about statistics or computing. And for those people, when I work with them, I think there is a gap in terms of communicating the main idea and in terms of how to make the project more meaningful and how to use better, more advanced statistical and computational methods. And I think in order to be very effective in the collaborations, both sides should learn from each other. So when I work with other people, I try to learn the domain knowledge in the corresponding field. And for the medical and the biologists, if they want to get the most out of the collaboration, I also think it's important for them to understand the basics of computing and machine learning aspects.
Thanks very much. And Professor Ant, please.
Yeah, I mean, I could add, I come more from the systems biology side, and this discussion has been, of course, quite an old one. It has been going on for a long time. In particular, in systems biology, you have a modular theorist coming, let's say, from physics and math, and they're now trying to do biology, and then they try to collaborate with biologists who don't really understand the math. So that's sort of an old problem in a way. So I would like to reiterate that it's important to know enough to have a good conversation, but in the end, I think, one can't be an expert in everything. So I do think it's valuable to specialize in a certain area, but I guess to know enough to have meaningful conversations. Yeah.
Yeah, thanks very much for your input. I fully agree that it's the obligation from both biologists and computational scientists to make sure that we understand both, we understand each other well, right? Okay, so maybe I can move the board to the audience. So are there any questions in any aspects? Please feel free to line up in front of the microphone.
Yeah, to follow up this question, probably I can ask something the audience would like to know, because for all your researchers, you are both reviewers and scientists, the person who writes the paper and who reviews the paper, right? I think also we want to hear from Angela's point of view. When we assign reviewers, when we have a panel with mixture of both computational scientists and experimentalists, or biology scientists, sorry for that, but how do we weigh the voice to make sure that both voices are heard, but we can still make a balanced and fair decision for authors? I have seen cases, for example, like when we assign both reviewers from both sides, the computational scientists probably support the work, but the experimental scientists may say, oh, this is lack of biological insights. What's your insight for these kind of decisions?
Well, I don't have any type of computational background, so I was glad to hear Robert say that you can't be an expert in everything, because I certainly am not. So my job as an editor is to sort of put myself in the place of the reader of our journal, and I want to see papers that can make accessible to that broader audience sort of what the significance of the paper is. So I don't need to get into the weeds with regard to, how the model is constructed, for example, and the architecture. So when I approach a paper, I'm looking to see that the authors have been able to convince me that what they have done is making a significant breakthrough that many of the readers will want to be informed about. And so, what that means in practice is that, generally I'm looking for some kind of application or demonstration of application of that particular model in a way that convinces me that, this will have broad interest. And for that reason, I will be, soliciting referees who will you know, even if they have no background in computational biology themselves, and they will tell me that, it's like, well, you know, will you be able to take this information and apply it to your own research? And, you know, I think AlphaFold was a very good example of that, because I cannot tell you how high the percentage of papers that we receive on the biology team now from all fields that have done an AlphaFold search and have some sort of AlphaFold, you know, reference in their work. So that's the type of papers that I'm looking for as an editor at Nature. You know, so basically something that will have a very broad interest and impact.
Thanks very much, Angela, for this very wonderful insights. Now I see some questions from the audience. Please go ahead.
Yeah, thank you. I'm from the Westlake University, and I'm thinking that as we know that the basic concept, the basic thing in the biology, that the sequence determines the structure and the structure determines the function. But for the, for this sequence, this amino acid sequence and structure, we have 3D structures. They are unified and they can, they do not have so much forms. But in terms of the functions, we have a lot of ways to represent the function. Do you think that are there some kind of unified forms can represent the function that we can? Or the general question is that what is the function? What the definition of the function? Would it be the text, the natural language? As is it possible to make the natural language as a universal representation for the function? Yeah, that's my question. Thank you.
Thanks for the very technical question, and please feel free to answer.
Yeah, maybe go ahead. It's an excellent question. I also often wonder about this. I think the Go terms and the easy labels, like the Ensign Commission numbers, they did a great effort trying to put a hierarchy and a definition for every function, for every protein that we know. But it's still very, very challenging. Often a protein may be doing several, for example, catalytic reactions, like we know from the Frances Arnold work that they may have secondary reaction. And it's very hard for us to annotate those, right, as scientists. So I think that's the next frontier line, especially also in design and annotation. Like how do we have now great tools to have millions of sequences. We can use metagenomics, we can blast them, we can MSX them. Also for the topology, we can assign them to false. But then the next frontier is how do we assign them a function that is that has a nomenclature that is understandable for every scientist. That's going to be a big challenge. I agree.
Yeah.
Yeah, great answer. I think about function a lot, and then because we have projects that care about function, and whenever I meet scientists and they talk about function, I always ask them what's their definition of function, because I feel like everybody works in a different framework of function, and I'm getting to grow more to the idea that there is no functional label. I think it's a continuum, it's an infinite, because you could imagine that you have an enzyme that has a certain KMK cut. Now you have a very similar enzyme that has a slightly higher KMK cut, but it can have really big functional implications for that organism as a whole, right? So, and then how do you assign that? It's like, and then also the multi-purposeness of things, right? So if I find it is one of the hardest problems to make progress in function is that we cannot easily find a good definition that everybody is happy with. And I think somehow we have to think about how to do that. I have no answer for it. I was in a meeting where many smart people were for 2 or 3 days just discussing the topic, and at the end, everybody ended up with more questions than with any answers. So I think it is a big problem.
Yeah, thanks very much for the question as well as for the excellent answers. And in the meantime, I also encourage you to ask questions, because as we're approaching the end of the conference, maybe it's the sort of the last opportunity that you can interact and also communicate with our excellent speakers. And in the meantime, I have another very general question, maybe in a more historical developmental sense, since I believe AI technology, AI augmented biology has been around since around a decade, and then maybe even longer than that. And I would like to know, in your opinion, at what stage right now is AI biology now or has been evolved to? Because I believe that, with this question, my concern is whether you think it is still at a very preliminary stage, although with so many breakthroughs and so many developments in terms of technology already happening, or you believe it's already at a quite mature stage and it's about to bring a lot of more significant impact to biology. And of course, you may elaborate it based on your discipline, because I understand there might be different kind of situation in different kind of subfields. Yeah.
I mean, I can't answer the question a bit so in particular, but I mean, also I'm very mesmerized actually. As you say, the field is very old. It basically is as old as computers. I mean, it started in the 1940s and 50s with the perceptron, the single neuron, and happy and learning and so on. And even the learning algorithm, you know, the learning algorithms we use is basically from the 80s. So, and now we're sitting here 70, 80 years later and discussing the topic. I mean, I find it super fascinating. Yeah? But yeah, I don't have a good answer, but maybe someone else.
One area of tension that I still see in this field is that, you know, the power we have now for structure prediction is amazing. And at the same time, I try to reconcile that with what we know is happening in the cell. So when you think about, you know, a protein being made in the cell, it's being translated. And we were always sort of told, or it was assumed, that, you know, domains would fold as they emerge from the ribosome, and then the domains would sort of assemble into the final 3D structure. But when you look at what these models are doing when they are taking a sequence and folding it into a predicted structure, basically it's all the protein all at once. And so to me, there's still that tension between understanding what's going on within the cell and how these models are actually getting to the same point from a very different beginning. So I think that's a really interesting question that we don't quite understand yet.
I work at the interface of spatial omics, digital pathology, and precision medicine. And I can speak from my perspective for the field that I work on. And I feel that AI, although we are seeing a lot of applications of AI in these different areas, but it's still very preliminary. So in the last few years, we have seen in the the work on pathology imaging foundation models. And these have been the foundation for many of the other works that people are using. But for spatial omics, it's still at the very, very early stage. The part of the reason is that such data is so expensive to generate. And although people are trying to use the existing data to build foundation models, but it's far from enough. I think there's still a long way to go for spatial omics. And eventually, in order to really advance precision medicine, I think different modalities and the different aspects need to come together. And there's a lot of room to generate more data and learn more from each other and across the different disciplines and modalities. So I think it's at the very early stage, and there are many challenges that we have to overcome and think about the connections of different data types and different disciplines. So there is opportunities for the young folks in the audience.
Yeah, thanks very much. And any additional inputs? Yes, and yes, or maybe also,
maybe one critical one. I think we have a lot of AI progress, and it's really rapid. And I think you can see more AI papers now coming out than non-AI papers in bioinformatics, at least. But I think we have to be also very careful as a community. Many of these things become really hard to reproduce. One issue of it is compute. One issue is that somehow data is not clear and performances are not always holding up. And there is also a lot of memorization in these models. So I think there is more and more things where we actually have to distinguish for. This is really a breakthrough because of it has learned something very fundamental and general about it, or it really has too much memorization and might not be as useful for all the topics. Just to put a critical voice into it, I guess.
Yeah, I mean, I think, yeah, I mean, I would also agree that there's a lot of extrapolation currently going on in terms of what has been learned. I think what was also iterated at the very beginning at the conference, I think it was a talk about AI biology, sort of the creative step, essentially taking very different data sources, let's say not only redesigning a protein or a genome, but also thinking about, how life emerged in the first from sort of first principles, extending biology to the physical environment and the chemistry in certain environments, maybe on other planets, and thinking about how life can have emerged. And this sort of creative step, that would be, of course, a huge milestone in terms of AI. And these kind of things I feel are still a bit missing. But yeah, I mean, lots of progress.
Yeah, great. Very excellent inputs, and also a little bit maybe input on my question. So maybe we say AI augmented biology is new. It's only that in very recent years we are more aware of its presence, but actually it's been around us. I mean, no matter the science and also the works, maybe it's new on a more applicational aspect, but the fundamental principles of it actually exist maybe many, many years ago, just as our speakers mentioned. Yeah, thanks so much for your input. And while I don't see further questions from the audience, yeah. Are there questions from the audience? Oh yeah, please.
Hello, thank you so much. My name is Bifon Momanyi. I'm from SUSTEC in Shenzhen. So I have one question. So right now, most AI models decode biology by fitting existing data. We all know that. But then life is an open, evolving system. So it's not just a simple fixed data set. So my question is, are we really truly decoding life, or are we redescribing what we have already measured?
Yeah, very great question. We expect the answers from the board. Since it's a very in-depth question, needs a little thinking.
That goes in the direction of memorization versus generalization. I assume that there is a certain extent to generalization in all of these models. They learn something from these models, but there is a lot of internalization of the data in itself. So I would assume both, and I think we are not really good in measuring really how much progress we have in this. How much do we actually push generalization? That's why these things like CASP and these critical assessments that are independent measurements, we would need them way more in some way to really know how general are these methods really, because obviously in papers often, there is some kind of you get nicer numbers than you often do when you then really use these methods, so it's not always as general.
Yes, and Professor Ant.
Yeah, it's an excellent question. I don't know if you saw a recent paper by what a language model can learn, and it was about the trajectories that planets, so the orbitals that they can take. So if you train the language model about the trajectories that they can take, that will fit perfectly. But once the models were prompted about the physics that they had learned nothing. So like the inertial, the vectors were making absolute no sense. So it was a big breakthrough in one of these AI conferences, and it's kind of like similar to what is happening also with language models or any big model trained on data sequences. Of course, they can extract the patterns that they have been trained to. Once we ask them other tangential questions about biophysics or how they fall, they just won't have a way to know that, and it's they have internalized how to generate new sequences or structures, but other more meaningful questions that they were not injected to the model with physical or chemical priors, they had no way to know them. So it's challenging for the problem for the future.
Thanks very much, and I think Professor Ant also has okay. Thanks. Yeah, very excellent discussion. And another question from the audience.
Okay, so this is Kai from Xi'an Jiaotong University. So the conference name is AI Augmented Biology. So basically means that we actually borrow AI technology and from computer science, from mathematics, and to biology. Right? This is a one-way street. So we carry something over. And I think I'm basically looking forward to something that bi-directional. So when we have only one direction means that, so we used to have a support vector machine, kernel technology, Right now we have the CNN and transformers. So all these kind of the AI technology from the other field, handling image, handling sequences, handling language. But biology is far more complicated. We have emergence properties, so we have all kinds of the regulation. So there's non-equilibrium properties that may not be fit for the current AI technology or current computer science technologies. So what's in your view and how can we stimulate and how can we help the field to develop new ideas, and not only that we have all the single cell spatial or language model that's submitted everywhere. So I think we have to think carefully. We have to stimulate discussion and new thoughts. Yeah, thank you.
Thanks. Hey, yeah, please.
Yes, it's an excellent question. I'm actually also thinking quite a bit about it. Your question reminds me very much, so the two-way direction, yeah? So AI informing, obviously, biology, because there are a lot of tools, and biology, I would say, it's already informing AI. But this is a question, there's this famous article by Joel Cohen from 2004. He wrote about, I think the title was Mathematics is Biology's Next Microscope, Only Better, and Biology's Mathematics, New Physics, but Better. So he talked about how the two disciplines informed themselves and developed further. And obviously AI has a lot of impact in biology, but clearly AI has been modeled after biology, right? I mean, an artificial neuron is modeled after the brain, Hebbian learning, associative memory, there was a Hopfield model from the 1970s. As you said, convolutional neural networks are modeled after the visual cortex. So that has been going on essentially, or swarm intelligence based on collective behavior in animals. I mean, a lot of these developments have been going on, but I hope it doesn't stop at this moment, and it will continue to inform new AI technology.
Yeah, I think collaboration is really important. We need to foster collaborations for people from different disciplines. There are a lot of computer scientists, statisticians that are trying to develop AI algorithms. But these people, many of them, they do not have the corresponding domain knowledge. So it's very possible that they develop the algorithms inside of their head, which are not practical, and the problems they're trying to solve are not important. This is why I think it's very important to have a forum for people from different disciplines to work together and brainstorm together, so that algorithm developers get to know what are the most important problems they should focus on, get the biological and the clinical insight from the people in the other discipline. And I can use myself as an example. It's very easy for me to come up with an algorithm, but when I see the results, a lot of times, I don't really know how to interpret. And now is the time for me to find a collaborator and work together with my collaborator, look at my results carefully, and have a meaningful biological discussion and interpretation. And once I get the feedback, I can get fine-tuning my algorithm and revise my algorithm, so that eventually the algorithms and the methods we develop are really biologically and clinically useful. But I think it's very difficult for people from different disciplines to come together. And I benefit from being a faculty in the medical school. So I constantly talk to different types of doctors. I can talk to cardiologists. I can talk to oncologists, nephrologists, pathologists. But for many of the other people, if they do not work in a medical school environment, it's very hard for them to find a collaborator that can teach them and inform them about the biomedical knowledge and the clinical knowledge. Maybe there is a possibility for us to think about creating some interdisciplinary conferences and invite people from different disciplines to come together and tell people what are the main challenges we are facing and what are the critical areas that we should focus on for the AI algorithm development.
Yeah, thank you, Mia. So if there are no other questions from the audience, and we would like to wrap up the discussion by summarizing I would like to hear one summarizing sentence from each of you about the future of AI biology. You can either recommend anything for early researchers or just share your opinion or your wishes for the future. Maybe starting from Angela.
So I fully appreciate that these models for structure prediction are just prediction. And so I think still what I want to see in the future is sort of a better understanding of the actual physics of protein folding. How do these models know what to do? I think there's got to be some answers in there somewhere, and I'm waiting for that paper.
Yeah, I think I can nearly copy that. I would really love to have more interpretable models so that we actually understand what they have grasped that we haven't grasped, and so that we can actually maybe learn more from them. I feel like that is something where we could profit the most from. So I hope this is going in this direction. I see more work happening, but we're still pretty much in the beginning on interpretability in these things.
Yeah, I think the future for AI in biology is very bright. As I mentioned earlier, I think the field is still very early on. And for computational tool developers like me, I have a lot of ideas. I can come up with many different algorithms. But what I do not really have is the enough training data. And I'm very excited about the prospect that in the near future, we are going to see an explosion of various types of data, and that will be the gold mine for computational researchers to develop useful AI tools.
Yeah, I think I'm going to say similar things to what Martin and Angela said. We see that these models perform really well across prediction and generation. So they must have learned something. But when we look at the papers over the last 5 years, we see that mostly methods were used as an evaluator, so as a predictor, or when we look at interpretability methods, they were used as to check whether they are actually learning through attention scores, to see whether they are learning what they were trained to do. It's a pity that we don't use whatever they have learned to teach us, and we hope that in the future we can see more work where we get biological and chemical insights directly from the model. And another point that I think for the future that we'll have to think about is what do we do once we run out of data? Because for PLMs or generative models in structures, we already hit the limit, kind of. So we are going to need to learn better ways to model the architectures, because so far we've been taking them as they are from the NLP and computer vision fields without minor modifications for the biological problem that we hadn't had, and also start injecting more rational processing to the training of the architectures themselves, so that we don't need the whole world of data, so that they also can interpret some chemistry or physics directly into the training process.
Yeah, I mean, I basically reiterate what I said a bit earlier. I mean, I would like to understand, or I would like to see more sort of creative leaps of novel understanding, and also teaching us what's behind these novel ideas. Basically working for AI on very complex, multidimensional, multimodal data, and having really big leaps of understanding and bringing us forward in terms of understanding stuff, or even having ideas, hypotheses we haven't really come up with ourselves. So that sort of leap I would like to see in the near future, ideally.
Yeah, thanks very much. And in the end, I would like to thank you again for your participation and for your wonderful insights, and also for the wonderful thoughts from the audience as well. Thanks a lot.
Okay, before we announce the poster winner, so we have selected 3 outstanding posters based on opinions from all the audience. And also, before we announce these prize winners, we will first give like a short acknowledgement, like how we write manuscript. For this conference, I believe that we have already had like a 2.5 intensive days. Thank you so much for staying with us and enjoy the insightful and fantastic discussions and talks. But I think I really want to acknowledge many people, because without their hard work and support, the conference cannot be so smooth and so successful. First, I would like really to thank Professor Zhang Chenyu's team and Professor Zhou Zhen again, and all the volunteers and people from Nanjing University who supported the conference. And they did a fantastic job to make all the workflow so smooth, and we thank them so much. And also, I want to thank my colleague from Shanghai office. I want to thank Annie, thank Helen, and also Aris, who is not here today. They give so much input and so much help for this conference. I really thank them for their input and for their support. Also, I would like to thank my fellow editors, Angela, Wenbo, Sunjie, Jay, Ophelia, Panjie, and Arunima for all the for all your hard work by moderating all the great talks and give all the support throughout the process. And finally, I would like to thank all of you again for all the fantastic 2.5 days. We had a really, really great time in Nanjing, and I hope you like the conference, and we will see you again possibly for the next time in the near future. And now it's the exciting moment for the poster prize winner, and I would like to announce the 3 people who win the poster award are Zhou Xibin, Zhang Shugang, and Dong Hongyu. Congratulations. Yeah, please come to the stage. Is Zhang Shugang here? Yeah. Let's give them a big applause again. Okay, thank you. And now I would like to announce that this is the end for the conference. I hope you all have a good trip back and also have a great weekend ahead. Thank you.